I'm waiting. Yeah, I will put this one like, oh god. Something wrong. Okay, so for my Tuesday, Thursday class, don't forget lab exam tomorrow. What's going on next Monday? Lecture exam, good. So it's going to cover chapters 5, 6, 7, and 8. We are dropping 4. We're not looking at 4. And the study guide is up, so you can start looking at your study guide, start gathering your notes, start studying now. We're almost done with 7, and we'll probably get through most of 8 today, so we're going to find timelines. Okay, so the last time we were together, we were talking about ways that we could kill different microbes. We talked about silver. We looked at the picture of the guy that was silver, had taken too much silver. That blue color, that silver color. Like I said, somebody had asked if that color reverses, and at this point in time, no. So I don't know, maybe it'll reverse eventually, but it's been a couple years and he's still blue, so still looks like a silver. So be careful with your silver supplements if you take them. And then you've got clothing, so this was our last slide, I believe. So the next thing we can talk about is when we're using cleaners, one of the things you want to keep in mind is that sometimes it helps increase the efficiency of a cleaner if we can reduce surface tension. So remember, when we have our microbial cells, they're negative, and if they get near something that has a positive charge, they'll stick or attract. So sometimes when I'm cleaning up a mess, I'll want to use a cleaner or soap to help kind of remove those charges and neutralize them to make it easier to get those cells out of the way. So soap is great at doing this. It helps break bonds apart. If you ever have like a minute of time, a really fun experiment to do, you might have done this, but get a bowl of water and sprinkle a whole bunch of pepper on it. Okay, so you have this pepper sitting on the surface of the water. And then get your finger, and this is great with kids to show them how to wash their hands with soap. Touch your finger into the water, and the pepper just sits there, sticks to your finger, no big deal. Okay, take your finger back out, dry it, and then put a drop of soap on the tip of your finger, or you can do it with a Q-tip, whichever you prefer. And then put your finger back in the water, and the pepper goes whoop, and it scoots to the edges. And it's avoiding the soap because the soap is breaking the bonds between the water and the pepper, which is pretty cool to see. So it's a nice visual, it's something that's really cool, and it reminds us why we need to use soap when we wash our hands. So we mentioned this also a really good mechanical method, meaning that if I'm washing my hands and I'm moving, that friction can help cool organisms off of my hands as well. I always laugh when I would tell my kiddo when she was young to wash her hands. You know, she'd squirt soap on her hands and then stand there like this. Like, that's not washing your hands. Like, you have to move them. If you've ever seen surgeons scrubbing, y'all ever seen surgeons scrub like that? That's intense washing, right? I mean, they're like serious. They're getting in there and the knuckles and all that stuff. Okay, other agents that we can use, quaternary ammonium compounds. These are in a lot of our cleaners. We find these a lot of times in certain types of like wipes, like Crock wipes, things along those lines. A lot of these are cationic, which means they're positively charged, and again, the idea behind this is they'll attract and pull cells towards them because the cells are more negative. They're really good at disrupting the plasma membranes. At least that's what we think. We're still trying to figure out exactly what they're doing, but we know based on our skin irritation with these that it's doing something with the plasma membranes. They're very effective cleaners for surfaces. The one limitation is they can't kill endospores. So we're leaving behind endospores. Again, it's kind of like trying to figure out how much risk that is. So for example, like if I have C. diff, I really do want to do something that's going to kill those endospores. But if I have something that's not an endospore form or in a room, it's going to be fine to clean with. So it's kind of like a case-by-case basis. So with our Ch.7, that's going to decrease surface tension. We have acid anionic detergents, so these are things that we can clean with our sanitizing. So anions mean they're negatives, they're going to disrupt cell walls. And then we have quaternary ammonium compounds, which are cations, which again disrupt the plasma membranes. So with this, these are some good cleaners that we can use. And again, I always say to read the labels of any cleaners that you have in your house because you might be surprised. Again, one of the big ones is letting cleaners sit. So a lot of times they need time to interact and sit with the cells before we let them on. Also, we have a lot of use for microbes as far as food preservation goes. So some of the ways that we can preserve preserved food is actually through product that is manufactured by the microbes themselves. We talked about how antibiotics were manufactured by microbes. Remember we talked about penicillin, coming from penicillium. The same is true with other preservatives. We've learned that there's things that microbes or molds produce that's going to inhibit the growth of the neighbors. And again, that has to do with competition. Now we can collect these products that they're making and put them in our food, and it's harmless to us. They break down the body, but they prevent microbes from growing in the food. So they help keep the food preserved. So some examples of this would be things like sorbate acid, calcium propionate, benzoic acid. These are all ones that come from different types of molds or bacteria. If you read food labels, you'll see them in there, and they're preventing bad molds and bad bacteria from growing. And usually they don't affect the flavor. If they do have any type of flavor to them, they tend to have a little bit of a tart flavor. So something that's not bad to us. It's something that's like a crisp flavor. Nitrites, that's another one that's been used, and you'll see this in things like hot dogs or sausages. It's used in a lot of meat or deli products to try to prevent CBOT. So we talked about Clostridium botulinum and how it can grow and cause food poisoning. So keeping those endospores from hatching out is a way that we can prevent CBOT. So a lot of foods have nitrites in them. There's a lot of research out there where the question is, are nitrites harmful to us? And that's been kind of something that's been debated a lot. From what we found in studies, low levels of nitrites are not harmful. But if we go super high, they could possibly be carcinogenic if we go super high. Now, it's always hard with these studies because you have to take everything with a grain of salt, because usually we're not testing on people when we're doing food studies. What are we usually testing on?
Animals.
Animals, and usually mice and rats, which are very small. So, for example, there was a study that came out about using aspartame, right? So aspartame is an artificial sweetener. And there was a study that published that said if you gave, mice aspartame, they would develop cancer. And so it was like this big scare about 15 years ago. People were like, we can't use aspartame, we're going to get cancer. And then when the study was looked at, they were giving the mice essentially about like a 24-pack of Diet Coke a day. So I don't know about y'all, I don't know anybody that drinks a 24-pack of Diet Coke daily. Maybe on some days you might have a bunch, but 24 every single day for months and months and months. So again, with nitrite, the same kind of study was done. It was like, oh, nitrite can induce cancerous cells, but it's with really high levels. So unless you're eating hot dogs every hour on the hour, you're probably safe, all right? So again, it's the kind of thing you kind of take with a grain of salt. Some molds produce antibiotics. We saw that with penicillium. So mantomycin is something that we see a lot of times added to cheeses because that also prevents bad molds from growing. And one thing that grosses my kids out, if I find mold on cheese, I just cut it off because mold is naturally in cheese. I don't worry about it. Oh, my chicken fries me crazy. My daughter and my nephews and nieces, they're like, oh my gosh. Well, I'm like, that's the fun of being a microbiologist, is knowing whether it's risk or not. Aldehydes. So these are other things that we can take out with antibiotics or with cleaners. Aldehydes basically are going to go in and they're going to break apart any cross-links that I have with my amino acids. Now, the reason that this is a big issue is if I'm breaking cross-links with amino acids, then that's going to affect things like my cell walls, right? Because remember, the cell wall is the peptidoglycan layers, but we have amino acid cross-links, right? So if I take them apart, those layers aren't stacking so well anymore, so it can affect the cell. We also can use aldehydes in things like a medical preserving. So if I take like formaldehyde and I put tissue in it, it prevents the breakdowns, the excessive breakdowns of like proteins and lipids, and it helps basically preserve the tissue as is. So that's something we can do. Now, we have to be careful with these because these are cancerous at high concentrations. There was actually a really popular hair treatment for a while called a Brazilian hair treatment or straightening treatment that was pure formaldehyde. They've fixed it since then, it doesn't have pure formaldehyde in it. But the way it would work is you put this stuff on your hair and then you would heat it, and the formaldehyde would change the crosslinks in your hair so you go from curly hair to straight hair. Or you could get rid of waves, it would be super, super straight. The problem with that though was when you would heat it, you would release a gas that running, they were having trouble breathing. Anytime that happens, stop what you're doing. Stop and walk away. Put the stuff down, walk away. And sure enough, they had some hairdressers that ended up with lung cancer from being exposed to these formaldehyde treatments. So kind of scary. And the other concern is that right now we're not seeing issues, but people who had early Brazilian hair treatments, they might end up with cancer of the skin of the scalp. We're not sure. We're watching it so far, no, but it's something we're watching. They do still offer this treatment. It does not have the pure formaldehyde in it anymore, so it is safer now. So just so you know, if you do see it on a menu or on the hair salon, it's safer now. And then the last slide we'll leave with is showing how resistant organisms are to all of the cleaning agents and all of the techniques to get rid of them that we talked about. So down at the bottom here is the least resistant, that means the easiest to kill, and at the top are the most resistant or the harder ones to kill. So endospores right up there at the top. We've talked about how endospores are pretty indestructible, they can live for hundreds of years, if not thousands of years, so really hard to get rid of. Above them are prions. We'll talk about prions when we get to our virus chapter. Those things are fascinating. Below that, mycobacteria. So what do mycobacteria have in their cell walls? Mycolic acids, good. So they have mycolic acids, which make them very resistant to any type of cleaner being able to get close enough to them. Cysts and vegetative protozoans, they're in a state, we'll talk about them in chapter 12, that are similar to endospores, kind of like an egg-like state, so it's hard to get through. Next, we hit gram-negatives. The reason gram-negatives are so hard to get rid of is because they have that extra LPS layer. That lipopolysaccharide layer protects them, so it makes them hard to target. They also have porins. What do the porins do? Yeah, they expel stuff, they spit stuff out. So getting stuff in there is, when we get it in, we can't keep it in, they spit it right back out. Fungi, usually we're looking at spores. Viruses we'll be talking about in a future chapter, chapter 13, I believe it is. And then gram-positive bacteria are ones that are pretty easy to kill if you take out that cell wall and they fall apart. Okay, the presence or absence of endospores has an effect on microbial control. But why are gram-negative bacteria more resistant to chemical biocides than gram-positives? What do they have? They have that LPS layer. Yeah, so just by having that LPS layer, it's an extra bouncer. If I target their plasma membrane, if I target their cell walls, they're still okay because they have this LPS layer, which is doing the job of both of those. So it's very hard to get rid of gram-negatives. So it's possible, but it is much harder than if somebody has a gram-positive infection. All right, and that will get us to the end of chapter 7, and then we get to jump into chapter 8, which is genetics. So you guys are still typing, so I'll let you type for a few more seconds. And you don't have to write everything on the slide, it's okay. You can just say LPS. I just don't want to do it too fast. All right, are we good? All right, so with that in mind, we'll leave chapter 7, and we'll start chapter 8. Okay, so chapter 8, we're going to be looking at microbial genetics. We're going to talk about what genes are, how our bacteria share genes, how we get some interesting mutations out there. So let's jump in. Do we have any questions from 5, 6, and 7 at this moment? Okay, let's jump in. So there's some terminology that's going to go with this chapter. The first term we're going to use and throw around a lot are genes. So we're going to talk about like the bacteria have genes. The bacteria have genes that can do this. All a gene is it's a part on a chromosome that's going to code for some sort of trait. So, for example, usually genes are coding for proteins. And it could be in us, for example, a trait like curly hair, or a trait like straight hair, or a trait like blue eyes, or a trait like dark skin. So we have all these different traits. And a lot of the differences that we see in humans is just protein based. That's all it is, whether or not we have these genes or not. We're going to learn that bacteria are a little more simple than we are because they just have a single chromosome while we have multiple chromosomes kind of helping drive our bodies. So we have a lot more input with our genes than they do. Now, a genome is all of the genetic the Genome Project that was in the news. And the Genome Project was looking to see if we could identify every gene on the human chromosome. And so it was like a great big project, all these labs are working on it. And we finally did map out all of our genes on the chromosome. And does anybody know what the average number of genes per human chromosome is? Our average number ended up being about 28,000. That was about our average. So anywhere from 28 to like 32, but usually closer to about 28,000. So that means we have 28,000 different genes dictating how we look, how we speak, how we talk, how tall we are, how short we are, all of that stuff is dictated by genes. So we were really excited by this because we thought, man, that's awesome. Like, we are so awesome. Look at all the numbers of genes we have. And then we started sequencing other organisms, and we found this crab. And we were like, oh, this is a simple crab. It's going to have less genes than us, and it had 140,000 genes. So then we were quick to publish a paper saying the number of genes does not in any way indicate intelligence, which I love. Like, we had to immediately publish that paper because we refused to admit that a crab with more genes might be smarter than us, right? We can't do that. And that's true. It turns out the number of genes really dictates a lot on how they're used. So we're going to have enzymes that kind of regulate our genes. We don't have to have as many because these single genes, depending on how they're cut or used, they can make different things. So just a little side note. Yeah. What's that? You want the previous one? This one? Okay, sorry. Did I get too excited there? I understand. No, you're good. Hey, if you ask, I guarantee somebody else is looking back right now too, going, then come and shoot us. Are you good? Alright. So with this then, the genome in our cells, remember, is DNA. DNA is always our boss. And when we talk about eukaryotes and prokaryotes, we always have DNA and RNA involved. When we talk about viruses, which like I said, we'll be covering in chapter 13, so coming up soon, we're going to be talking about them as being either DNA or RNA. So they don't have both, they have one or the other, which I would really like. Bacterial chromosomes, they're super simple. They tend to be in this nice circular loop. They're really small. So it is easy for them to be able to have mutations in their genes and to share genes back and forth because they have these really simple little chromosomes. For us, we tend to have linear chromosomes, so instead of being in a circle, they're in straight lines. And does anybody know what's the total number of chromosomes in the human body?
23 pairs.
For how many chromosomes in total?
46.
46, yeah. So we have a total of 46 chromosomes. So we have a total of 46 chromosomes in the body. So on those chromosomes, that's where we find those 28 to 32,000 genes are in those chromosomes. So yeah, so we have 46 chromosomes. When we produce our egg and sperm, we have 23 chromosomes in there, and then they fuse together to make the 46. And that's where we get the shuffling that we see in our population. We also can refer to this as genetics. Genetics is really fascinating. It's where you're studying heredity, so figuring out traits that can be inherited, traits that can be passed on. There's a lot of things that we can do now if we're looking for specific traits. For example, if we have a couple who are wanting to procreate, we can do a quick gene check to see if they have any traits that they need to watch for. You have your hand up, Leila.
Genes.
This one?
How come it's usually protein when it should be nucleic acid?
Okay, so what do the nucleic acids code for? Remember, we talked about how DNA is the boss, right? And DNA is going to tell our cell what to do, right? But DNA has to talk to RNA, and then who did RNA talk to for what the cell needed? The ribosomes. And so ribosomes make proteins. So proteins are basically going to be the doers. The chromosomes, the genes, are the blueprint. Okay, so the blueprint or the map of what we want to get to. The proteins are how we get there. Think about it that way. So think of like DNA or a gene kind of being like a blueprint. The protein is the lumber that everything else has got to put together to build the house. Does that make more sense? It's like the supply. Good question. And you'll see how it works, because we're gonna have that coming up in a few slides. So for example, in my ex-husband's family, cystic fibrosis ran in their family. It's a recessive gene. So he did not have cystic fibrosis, he was a carrier. So one of the things we did when we were ready to have kids, I went and had a blood test to see if I was a carrier. It was just gonna help us figure out if we'd have to possibly plan for a kiddo that might have cystic fibrosis. Luckily, both of my genes were good. I held that over him. I was like, yes, it's my good genes. So both of my genes were good, which means my daughter might be a carrier. She might have inherited this defective gene. I don't know. We'll have her check when she's ready to procreate. But for now, all that told us was that we were going to have a child that was not going to have cystic fibrosis, because I had two good genes and he didn't have one bad. So just something to keep in mind. So it's just sometimes nice to know that and take stress off of the family. So if we talk about an organism's genotype, that means all of the genes that they're carrying for different traits that they have. Their phenotype is what we actually see. Okay, so think of P for plain to see, phenotype is plain to see. That's what I actually see. So for example, going back to my ex-husband's example, where he is carrying cystic fibrosis, so his genotype is cystic fibrosis carrier. But if you were to talk to him, he has no signs or symptoms because he has a good gene, so his phenotype would be non-cystic fibrosis. Make sense? So the phenotype is what you see. Another great example of this is when we get to things like Punnett squares. Punnett squares are where we're able to kind of predict what offspring will look like. Now, Punnett squares are usually done when we have a mixing of genes. So these would be more eukaryotic than prokaryotic, because we're mixing two different types of genotypes together to get different phenotypes. So what I mean by that is we're mixing like a mother and a father together. Their genes are going to mix, and we'll see what their offspring is going to have. Now, how many people in here have used Punnett squares before? Yeah, they're pretty common in science class. If not, no worries, we'll go through it. The big things you need to think of is if you see an uppercase letter, that means that it's a dominant trait. So usually if there's an uppercase letter, that means dominant trait. If something's a dominant trait, then that means that I will see it in your phenotype. Okay, so I will be able to see that expressed. If something's a lowercase letter, then that's going to be referred to as a recessive trait. And that means that I might not see it in your phenotype. It could be hidden. Okay, so it could be a hidden trait. So for us, we inherit a gene from mom and a gene from dad. So let's say that we get a gene for a dominant trait from dad and a recessive trait from mom. I'm going to see dad's gene because it's dominant. And this could be vice versa. This is what we look at, is whether they're dominant or recessive as to whether they're going to have an impact. So in this example here, we can see that we have up here capital Y, which is dominance, which means that I'm always going to be yellow if I have two dominant genes. If I have a dominant gene and a recessive gene, I'll see the dominant phenotype. So I'm still carrying the recessive, but it's masked, okay? And then over here, if I have two recessives, I might see the trait that was hidden. Does that make sense? So two recessives mean I'll see a hidden trait. If I have a dominant, it will always go over that recessive trait. So that's referred to as our phenotype that we see versus our genotype. Now, the way that we can make Punnett squares really quick and easy is you just kind of make a little square and put some blocks in the center. Makes it pretty easy. You put the mom on top, typically the dad on the side, and then you put in whatever genes they have. Now, the cool thing about this, let's go to our expression here. Let's say mom has a recessive gene and a recessive gene, so I put her two genes here, and dad has a dominant and a recessive. We can make a Punnett square, and what the Punnett square shows us is the chances of the offspring having these traits. So this is my offspring chance, okay? So it's real simple to do this. Bring dads across. So capital, capital. Everybody see how I did that? I just kind of zoomed it across. Bring dads across again. Lowercase, lowercase. All right? So we're down with dad, so now we have to do mom. So mom, we bring hers down. Lowercase, lowercase. Bring hers down, lowercase, lowercase. So in this particular example, I have a 50% chance, right? Because each square is worth 25%. I have a 50% chance of my kiddo getting a dominant trait, or a 50% chance of my kiddo getting a recessive trait. Everybody see how that works? So that's a Punnett square. And I bring that up because it is important when you take classes later in your medical school or whatever training you're going into, they'll talk about like different genes that people inherit and things like that. It's nice to have these Punnett squares in their head. So one of the ones that comes up a lot, and this one's actually more involved than it originally appears, is something like the Punnett square or eye color. If I have a capital B, let's say I'm going to have brown eyes. And again, this is more complicated than it sounds, but we're going to keep it simple. If I have a lowercase b, I have blue eyes. Okay? The reason why brown eyes are more common in the world is because it's a dominant gene. Blue eyes are recessive. All right? Actually, blue eyes are defective. We're not making proteins in our eyes, so that's why they tend to be so light. So, some people are like, oh, I like blue eyes. They're not working right. So there you go. Just gotta say it. Okay, so is it possible for two brown-eyed parents to have a blue-eyed child? Yeah, it is possible, yeah. So we see this a lot, and this is something where, like, when this happens, you can imagine it creates some tension in the delivery room, right? I mean, yeah. Also, another thing, have you ever seen a child who was born with light eyes and then they got dark?
Yeah.
So that's protein development. So when they're first born, a lot of times babies who are going to have brown eyes will have blue or green eyes initially, and then as they age and proteins start to invade, they start to get the darker eye color. So eyes can change a lot. Here's an example of how we can have two parents with brown eyes having a blue-eyed child. So same idea that we saw here. I bring mom's genes down on both sides, dad's genes across on both sides, and what we see is we have a 25% chance of having a blue-eyed kiddo. Now, it's a little more involved than that. There's actually about 19 different proteins that dictate eye color, which is why it is not simple like blue, green, and brown. Have y'all seen all the different colors of eyes? There's like hazel, there's all different shades of green, all different shades of blue. The different shades have to do with this other 19 proteins kicking in and making different variations, which is so cool. I love all the variations that we got. Now we're going to do the PTC tasting in lab, because I'm actually going to have you do that one. Has anybody ever done PTC tasting? You do? Okay, great. Shall I tell you my horrible story about PTC tasting? We always like stories, right? Okay, so first of all, let's talk about PTC tasting. So PTC is looking for a gene that we carry in our saliva. And it's a gene that if it have a dominant gene, we give you this paper, it's like a strip, and you put it in your mouth. And if you have a dominant gene, you get a bitter flavor. Okay? You get a bitter flavor. And I have to tell you, I have no idea what it tastes like because I'm recessive. So the first time I took a PTC test, and again, we'll do this in lab on Wednesday and Thursday. It's a little strip you put in your mouth. If you have the gene present, it breaks the enzyme and you get this bitter release. If you don't have the gene present, it tastes like you just put a piece of regular paper in your mouth. You get absolutely no taste whatsoever. So in my first class, when I did this in biology classes back when dinosaurs were here, my lab partner and I were working together, and she was like, do you think it's like, what do you think it's gonna taste like? I was like, oh no. And she's like, well, you go first. So I was like, okay. So I put it in my mouth. We didn't know I was recessive. So I'm like, I think it's a trick. I was like, there's nothing on it. So she's like, oh, okay, cool. So like I spit my mouth. I was like, yeah, there's nothing on this. So she puts it in her mouth. She was a double taster. Super bitter, super strong. So she like puts it in her mouth. She's like, oh, and she's like running out of the room, drinking her water bottle, washing out her mouth. She said she had this bad flavor. She had to get some gum to get rid of the flavor. It was like super intense. And I'm looking at her and I put another piece in, I'm like, I don't taste anything. And she's like, no, it's really bad. So I thought she was pranking me, she thought I was pranking her. And then finally our instructor explained it was because we had different genes. So that was kind of a cool thing. So with PT, if you're capital, then you're a taster, that means you'll get that bitter flavor. If you're recessive, then that means that you have no taste. I have no taste whatsoever. Okay, so let's try to figure out a Punnett square here. So let's say that I have somebody who is recessive, okay, and they're with somebody who's recessive. What kind of offspring could I potentially have here?
All recessive.
All recessives, yeah. So in my other horrible story about PTC is we were doing open house at Cal Poly University. And I was an undergrad, and I was helping out. We were doing all sorts of science displays and science demos. And one of the ones we had was PTC tasting. And it seemed harmless. It was cool. You talk and put a piece of paper in your mouth and see if you taste this flavor or not. And we had a couple come up, and the woman put the paper in her mouth, and she goes, I don't taste anything. Or like any kind of aftertaste, anything. She's like, no. So there's her genotype. She's double G, recessive. The guy with her puts a piece of paper in his mouth, he tastes nothing. Okay, so again, double recessive, cool. Their child puts a piece of paper in their mouth and goes, ew, yucky. Yeah, some of you are laughing, you're kind of getting it, yeah. They're recessive. Could that kiddo actually taste anything if they were both the parents? No. No. So that means there's, a fox in the chicken house here. Something went wrong, right? So somebody gave this kiddo a dominant gene. All right? Because if you have even just one dominant gene, you get a taste. So we're looking at this kid like, oh, please be a nephew, please be a friend's kid, please don't be their kid. And sure enough, the dad kind of looks at this and he goes, wait a minute, you tasted something? And he goes, yeah. So they all try again, and the mom's getting this look, and we're like, oh crap, we're all looking for an adult, because we know where this is going. We're only like 21, we're not old enough for this, we need adults now. We're looking around for our teacher, he's not there. So sure enough, the kiddo again says, yucky, we're just like, oh no. So we try to show him a different display. We're like, oh, come over here, look at this display. The dad's like, no, I want to look at this one again. I want you to explain the Punnett square to me again. We're like, we don't want to. We're done with this display. We're moving on. Yeah. So they walk away. A little time goes off, and then you hear yelling in the sacred halls of Cal Poly Fisher Science Building. Well, I didn't know if you were the father or not, and you asked me to marry you first, so I said yes. And we're like, yeah, we're looking around. Oh, the drama. Yeah, they're full on going at it right there, Fisher Science Hall. It was just. I didn't do this test in my classes for a long time. I was a little traumatized. I have brought it back now. The last time I handed out the strip of papers after telling the story, one of my male students goes, can I take it home to test my kid? I said, no. I don't want to know, and you don't want to know. We're not doing that. So anyway, eye color, PTC, they're just used to show the different genes that we have, the different proteins. Yeah, still have trauma over that story. Okay, so going back, what is a gene? What is a gene mean? B. Everybody agree? Functional protein? Yes. If you ever doubt, always go for the longest answer. don't do that on my test. I had one instructor that was the truth. Like you didn't even have to study, you just pick the longest answer. Because he would always have one answer that was really long, guaranteed that was the right one. So I had another instructor that was always C. You ever had that? Like they tell you, if you're not sure, pick C. For some reason, instructors like that. Don't do that on my test either. True or false, viral genomes can be based on DNA or RNA. True, yeah, they don't have both. So they're kind of oddballs. Again, they're packet light. So if a virus comes in with DNA, we're going to make their RNA. If they come in with RNA, we're going to make their DNA. So they make us make whatever they need. Kind of fascinating. Okay, so again, genome sizes can vary across the board. So we have about, the smallest virus has about 4 or 5 genes, so that's pretty tiny. E. coli has about 4,300 genes. chromosomes do we have, although usually I have better, it would be extra credit also, but usually I have better extra credit. E. coli, even, you guys know how small E. coli is, we look at it in the lab, how tiny it is, it's so hard to see with a microscope. If I took their genome and stretched it out, it would be the total length of 1 millimeter, which is about the thickness of a credit card. So not very big. I mean, think about how tiny that little genome is. That's with almost 4,300 genes. That tells you how small these genes are. For us, with us having 46 chromosomes and our 31,000 genes, like I said, it's anywhere from that 28 to 32, so 31 is the one I put on here. If we stretched our entire sequence out, it would be a total of 6 feet. So I'm 5'3", so we will somewhere right there, 6 foot. All right. So very long, we stretch them out bigger than the cells they're in. Now remember, DNA is the king of the cell. It's the one that has the blueprints and all the information. But really, what it's doing is it's using that information to help drive cellular functions. The workers, the ones that are actually going to be making the product the DNA wants, are the ribosomes. They're going to make the proteins. And again, our interpreter, the one who sits in between the ribosomes making the proteins and the DNA, is RNA. So RNA is always our interpreter who can speak between the two. So all organisms follow this basic pathway to be able to make product that they need to survive. So our central dogma, as it's referred to, is a pathway that's done by E. coli or humans or even that crab that had some genes. And what this pathway is that we go DNA to RNA to protein. So we always, it's kind of like a one-way street. So you gotta go one way. And again, that's referred to as the central dogma. So it's like this pathway that every cell on the planet is gonna follow. So with this in mind then, the central dogma was something that was under a lot of controversy when we had our new Pfizer vaccines come out for COVID. So remember, the controversy behind the COVID shots was that when they were doing injections, they were giving us proteins, right? And the concern was, well, if we get these proteins, they'll mutate back into the DNA, and they might change our DNA. But based on the central dogma, that's really not going to happen. It's kind of like going to San Luis Obispo, all the one-way streets up there. It's like you can't go the wrong way. Well, you shouldn't. You shouldn't go the wrong way on a one-way street. I've seen people do it. You shouldn't go the wrong way on a one-way street. And that's kind of what would happen. That's what some of the concerns were, is that if I gave somebody a protein, it would try to go backwards. But the risk of that was nil. It really wasn't possible. It didn't have enzymes. There are certain enzymes that would be involved that would be able to go that way. So that was some of the controversy around it. If you did get the shot that's going to make you feel better, don't worry. It did not affect your DNA in any way, shape, or form. All right. Now, the central dogma works together in a process called transcription and translation. So the idea behind this is DNA has the message. RNA is going to go in and transcribe. So transcribing means to kind of like write down the message, like a secretary would transcribe. And then the RNA is going to take that message that it wrote down to the ribosome where it's going to be translated. And when we translate it, we make our protein. All right. So we're carrying the message and we translate it. This process is called gene expression. So if I'm making a protein, then I'm expressing it. Okay. So the idea behind this then with gene expression is that we are going to the ribosomes and we're making a functional product, the goal, which is the protein. Now, to help us in this process, we always have to rely on RNA. And there's different types of RNA. So the first RNA that's involved in this process is the one who's going to carry the message. And so it's going to be called messenger RNA. That makes sense, right? So it's going to be transcribing, writing down what the DNA wants. So it does this by matching base pairs with our DNA. Once we take this information, once we transcribe it, we can then share that information later on with the ribosomes. So transcription is a pretty straightforward process. So we'll have a strand of DNA. Draw out some bases for me. Give me some DNA bases. Here or so. Okay. just give you bases, A, T, C, G's.
Oh. A, T, G, Z, A.
Okay, T will go with that, so 3, 3, 3, okay, perfect. So the way this works is messenger RNA comes in, and it's going to match up base pairs, alright? So what typically is going to bind with an A? T. Now does RNA have thymine?
No.
No, what does it have instead?
Uracil.
Uracil. So wherever there's an A, my binder is going to be a U. Make sense? Okay, then we go over to T, thymine. What usually binds with T's?
A.
A, so put an A here. And we go over to our cytosine. What usually binds with cytosine? Guanine, so we have our G. So the way we do this is we just build our strand. What am I doing here? Okay, got it. So we build our strand based on what's in the DNA. Think of this as kind of like a recipe. The DNA is holding recipe cards, the mRNA is transcribing them. Okay, it's like when we share recipes. Now the way they're always going to do this is 3 bases at a time. And these 3 bases are referred to as codons. Okay, that's a term. So a codon is a 3-base sequence. And these 3 bases, in turn, are going to be responsible for making an amino acid. Okay, so my codons, I'll give it the darker color here, are going to be later on transcribed into making an amino acid. Everybody with me so far? Okay. If I gave you a sequence of DNA, could you go to mRNA?
Yes.
Okay, good. That's what I wanted to hear. Okay, so this process then, with messenger RNA reading the DNA, is going to start at a certain sequence of the DNA, which is called the promoter. And the promoter is basically like a start here card. We call that initiation. Okay, start here. And then what's going to happen is the mRNA is going to keep reading the DNA for a while. We call that elongation. It's going to keep reading 3 bases at a time. And it'll keep doing that until it hits a special codon called a stop codon. And the stop codon is like the end. So think about it this way. Think about the DNA as being like a story. How does every fairy tale start? Once upon a time. So we start with once upon a time. Once upon a time is going to be my promoter, my initiation. And then I read, and how does every fairy tale end? Happily ever after, the end, right? So the end is going to be my termination. Okay, so I got these codons kind of built in. Now, as with everything in life, there are enzymes involved. So we're going to have an enzyme called RNA polymerase. So what this means is it's going to help produce a copy of mRNA by matching the bases to the DNA. Okay, so what this RNA polymerase does is it helps read the DNA bases to make sure I'm getting the right base matching. Okay, so if I try to stick a C here instead of a uracil, my RNA polymerase is going to go, that doesn't fit. I want to have a uracil. Okay, so it's going to be like our checker, which is nice. It's like our proofreader. Now, where we start the process again is going to be at the promoter. That's like the, once upon a time. That's the sequence of DNA that says start here. And then we'll elongate and read along that and keep making extra copies of our mRNA and building our strand. Now, again, just as a reminder, in the mRNA strand, cytosine always pairs with guanine, so C with G's, and adenine is always going to pair with uracil in my mRNA strand. So if I was to give you 2 strands on a test, would you be able to tell the difference between a DNA strand and an RNA strand? Yeah, so look for those thymines or look for those uracils, right? So if I see uracil, that's RNA. If I see thymine, that's DNA. So those are huge clues. All right. So,
Can you go back? Sorry.
no,
Thank you.
that's it. Really fast. All right, so I'm going to build along the strand. So my elongation will take some time depending on how many amino acids I want to string together. So remember, every codon that we see here is a different amino acid. Now, amino acids get linked together to create what biomolecules?
Protein.
Proteins. And remember we talked about proteins, how they can come in different sizes, right? Remember we talked about how proteins can have primary folding. That's like the early lines of Disneyland. Secondary folding, that's when we're starting a coil. Tertiary folding, that's where we're folding on top of our coil. And quaternary, where we're bringing a whole bunch of strands together, okay? The same is true here. So I'm gonna keep building a strand until I reach the end. So my ending, where it says the end, is referred to as the terminator. That is boring. We have to say it like Arnold Schwarzenegger. We have to say the terminator. It was a horrible accent, but wasn't that fun? Yeah, so I want you to say that terminator. And that will be where we're gonna stop, okay? So I'm gonna hit a sequence in my DNA, and basically what the terminator has happened is I'm not really able to bind with it well. I can't bring in bases, so then I'm gonna stop. Okay, so the terminator says stop here. I don't have mRNA to match up with it. That's basically what's gonna happen. So at that point in time, my strands will separate. DNA, which had unfolded so the mRNA could read it, coils back up. Okay, remember DNA is usually like a double helix, right? So it's a coil. So it unfolds during this process to read and then coils back up. Kind of like opening a book and then closing it, okay? When I close the DNA, the mRNA falls off. It is now its own strand. It's ready to leave and go to the ribosome. Okay, so it's gonna get ready to go to the ribosome. Okay, the next process, which is translation, this is where I'm reading the message, this is gonna take place in the ribosomes themselves. So remember, prokaryotes and eukaryotes have ribosomes, which is where we're going to make our proteins. So the way we're going to make our proteins is we're going to read the mRNA that arrives. So this mRNA strand is going to arrive at my ribosome, and my ribosome is going to start reading it a codon at a time. And each codon is going to be creating another amino acid. Okay? So we're going to get each protein that comes in. Now, the way this process works is the mRNA arrives at the ribosome, and the ribosome is going to have another RNA molecule that comes in to match the mRNA. Okay? So this is referred to as tRNA. So tRNA has the job of matching mRNA. Okay, so let's do a tRNA strand down here. All right, so the way this works is I'm matching my bases again. Okay, so if I have a U, what binds with U? A, good. If I have an A, U. If I have a G, C. If I have a C, G. Okay, you guys get the idea. All right, so now I have created my tRNA. My tRNA is referred to a lot of times as the anticodon. What that means is it's the exact opposite of mRNA. I like this. Doesn't this sound like some sort of like sci-fi movie? We've got terminators and anticodons. Like, how cool is that? Like, there's all these, that's like a villain's name, right? The anticodon, that's what it's saying. But all it means is the opposite of the mRNA strand message. Okay. Now, the reason tRNA is important is because tRNA is carrying a buddy. Guess who's riding on the tRNA? An amino acid. So this is where we're going to start actually building up our proteins. When tRNA arrives, it's carrying with it amino acids. Okay? So if I match up to the mRNA here, I'm going to be bringing in an amino acid that this mRNA dictates for, and I'll leave that amino acid behind in the ribosome. And then I'll go to the next one, and another tRNA arrives with its amino acid, and so on, and I start building a strand. We're going to see this in pictures, so don't worry. If it's confusing right now, it should be. We'll want to see some pictures. Okay. Now, before we go into some more pictures, just a couple of things you keep in mind. The genetic code is universal. So what that means is every single living organism is going to be reliant on DNA and RNA functioning in this matter. Okay? The differences are the code themselves. So the sequence of A's and G's and C's and T's are where we get differences in whether you're a human or a mouse, the base pairing is different. But the DNA, the mRNA, and the tRNA, that process is the same. Okay, so we're going to be following the same process. Now, with our codons, the codon that comes in is going to bring with it a single amino acid. So every codon that's out there has one amino acid associated with it. Now, as an amino acid, I might have several codons. We're going to see a picture that's going to explain this. So I have several codons that can code for me, but each codon can only have one amino acid. I know it's a little confusing, so let's see how this goes. So for example, one of the amino acids listed here is valine. And by the way, you will not have to memorize these. Okay, if I ask you to do anything like this, I will always give you a list of the amino acids. We're not memorizing the 20 amino acids. Okay, you'll do that in biochem. All right. So here's valine. And here's an example of all of the codons that are going to code for valine. Now, if I was to look at just this codon, GUG, that codon can only carry valine. It can only carry valine. But valine can be found with GUG, GUA, GUC, and GUU. Does that make sense? So I have 4 different codons coding for the same amino acid, but each codon is specific for that amino acid. Everybody okay with that? Okay. I know it gets a little confusing. Okay, so here is a genetic code table that should terrify you when you first see it, but actually we'll go through it. It's actually pretty easy. And if I was to ask questions on an exam, I would always give you one of these. All right, so let's learn how to read it, because this has a lot of information in it for us. First of all, the most important thing to do is to see if the genetic, is to see if the genetic code table is in mRNA or tRNA. Okay, so at the top of this one it says mRNA. The reason I bring that up, I had a student try to pull up one of these tables on Google, and they came to me and they said, your table's wrong, it's all different. And I looked at their table and they were looking at a tRNA table, which is the inverse. Same information, it's just different bases. Again, you're not memorizing it, I'm just letting you know, in case you look it up, like, look at the top. Okay, so let's see how to read this, it's so easy. These are separated into codons. Codons, again, have 3 bases. 3 bases. Okay, right here, this says first position. So what this means is that every letter from here and here and here and here, so in the box right by first position, is going to be the start of those codons. So for example, here's my G box, and every letter, reading across here, starts with G. Everybody see that? That's my first position. Up here, every codon listed starts with A. Okay? C, and then U. Make sense? Okay. Then at the top, it says the second position. So up at the top, I have the column U in the second position, which means every single codon listed in these boxes should have U in the second position. Make sense? Over here, everything listed in these columns should have C, A, and G. Everybody with me so far? See, it's not hard, right? And then the third position, it's telling you what's attached on the tail end. So here's U. So everything in this row is going to have U as the third position. Here's the C. Everything in this row has C. All right? Now the reason I'm showing this to you is if you're trying to find a codon, you know how to find it quickly on the table. Make sense? Okay. Now notice there's a couple of codons on here that are in white. So we have some stop codons. The stop codons are the terminators. Okay? Those are codons that there is no amino acid for them. So when my tRNA tries to bind with mRNA, I don't have anything that can bind with mRNA that has those codons. So all my tRNAs are swooping in trying to bind, and nobody matches those 3. So that's my sequence for time to stop. Okay, that's my stop codon. Now notice there's also one in white that says start codon. That's our AUG. It codes for methionine. Now, that's a special codon. The reason for that is the way I did the sequencing here, as an example, was kind of incorrect. I should always have to start with an AUG, okay? Because that's my start codon. That has to do with over here, when I was talking about how we have 28 to 31,000 genes, and I said it has to do with how we clip them or pick them. The start codons that always say like start here for mRNA are always going to be AUG. That's the once upon a time. Now, if it's AUG for mRNA, what is it for DNA?
TAC.
You got it. TAC. All right. So that means that I need to find. I have to read my sequence until I find that, and that's where I can start. Okay, are we good so far? Okay, I'm just making sure. Now, again, if I ask you questions on the test, I would give you a table like this. And you would be able to find the sequences. Maybe I give you a strip of DNA. I have you make some mRNA, and then read a sequence and tell me what amino acids we make. As I told you, we don't have to memorize. So for example, looking on here, I have AUU, AUC, AUA, AUG, or sorry, not AUU, AUU, AUC, AUA. That has ILE beside it. I go over here, the only one that starts with an I is isoleucine. So that would be the amino acid that would go there. Everybody with me on that? Here I have MET. I go over here, that's methionine. Everybody see how this is working? So I would give you a table that would tell you what the amino acids were. So in the past on exams, I've given a sequence of DNA and said what amino acids would match up with this DNA. So you would have to convert to mRNA, and then you would have to read a table to see what amino acids were related to that. Make sense? So we're not memorizing, we're using. Okay, any questions on that? Okay, so let's keep moving forward. Okay, so translation, the process of reading our mRNA is always going to start at a start codon, which again is AUG, which means that our first amino acid is always going to be methionine. Translation is always going to stop at a stop codon. That's where we fall off. So if we look at that table and we count up all the combinations, there are 64 potential combos of mRNA. These 64 potential combos can be used to code for the 20 essential amino acids. Okay? Again, that's because there's some that'll repeat again for amino acids. Out of our 64 codons that are possible, only 61 can actually make a functional amino acid. The other 3 are stops. Okay, so only 61 can make an amino acid. So what that means, don't worry, I'll go back to the slide, is if I go here and start counting the number of codons, I would have a total of 64 codons on here. I had to see big count of them, they didn't believe me. Trust me, 64 codons. I have no reason to lie. Alright? And then 64 minus 3, because I have those stop codons, mean that I'm only going to have 61 codons that are functional. Everybody good on that? I'll make sure. Okay. And again, we have a total of 20 essential amino acids. Okay, so let's go through our process. So the translation process is going to happen in the ribosome itself. And again, it's going to do a similar process to what we saw when mRNA was reading DNA. We're going to start with initiation, we're going to have elongation, and then we're going to have termination. So we're going to get to see how ribosomes work, and the cool thing about it is you're going to finally get to see why ribosomes look like a hamburger bun. Remember how we talked about how they have a big piece and a small piece? We're going to get to see why in the upcoming slides. So remember, here are ribosomes. We talked about how they have the hamburger bun appearance, the big and the small. We said for eukaryotes, what size of ribosomes did we have?
60.
We had 80, good. So we had a 60 and a 40. What about in prokaryotes?
70.
We had 70, we had a 50 and a 30. Okay, so just keeping that in mind. So these units come together. Now, the way the ribosomes usually are in our cells is they're close to each other, but they're a little separated. Okay? Yes?
Can we go back to the translation slide, please?
You got it. So the way the ribosomes are is they're close to each other. So picture like a hamburger bun close to each other. when mRNA arrives, the ribosomes are going to clamp together. And guess what's going to be the patty or the meat in between them?
The mRNA.
mRNA. Yeah, the mRNA is going to be the meat that's in between the two buns. All right, so they're going to snack together and the mRNA is trapped in between. That's one of the reasons why our ribosomes are kind of separate. So that way they can snack together, hold the mRNA in place, and then they can pull the mRNA through, reading it a codon at a time. So now that makes a little more sense, right, where we have two separate pieces. Okay, so this is what we're picturing in our head. So if I was to look inside that ribosome, so I go one here inside, I would see that there's three main sites associated with the ribosome. So now I'm looking at it inside. There's the A site, the P site, and the E site. Easy way to think of these. A for arrival. This is going to be where the mRNA enters in to the ribosome. Okay. The P site is going to be for protein. This is where I'm going to put the amino acids together to make my protein. And then the E site, if I'm leaving, what do you think E would stand for? Exit. Yeah, the E is going to be my exit sign. So an easy acronym, you can just look at this and say it's an APE, APE, arrival, protein, exit. Okay, so that's what we're looking at here. So what that means is mRNA is going to come in, and I'm going to read the mRNA a codon at a time. How many bases are in a codon? 3. Good. So I'm going to read 3 bases here. Now at the A site, this is where I'm going to be matching a tRNA up to an mRNA. Okay? So I have tRNAs floating all around, and remember what's attached to each tRNA? Amino acid. So they're floating, they're waiting nearby. mRNA comes in and they start swooping in, and enzymes try to grab the right tRNA that's going to match. When they match, they're going to bind together. And then together, the mRNA and tRNA are going to move from the arrival site to the protein site. Okay, that's going to be what we're going to be looking at. So here's a picture of what tRNA looks like. Kind of looks like a little hairpin, like a bobby pin. So at the bottom, I have the bases that are going to match up to my mRNA. And then there's my little buddy at the top, which is the amino acid. Okay? So I have this base sequence, this codon, that's going to match up to the mRNA. And if I match up, I'm now bringing in the correct amino acid that my DNA wanted in the first place. Okay? So this is my process that's going to be taking place. So that right there is showing the anti-codon. And again, anti-codon just means opposite of mRNA. Okay? Okay, so let's go on to the next one. So, in my ribosome, the first stage is going to be initiation, just like what we saw when we were making our mRNA. The mRNA binds at the A, or the arrival site of the ribosome. It's going to find the AUG site, our start codon, and it's going to match that with the tRNA that matches with our AUG. So if I have AUG for my mRNA, what would my tRNA be? You got it, UAC. So always be looking, if you're looking at tRNAs or mRNAs, because they're always the opposite of each other. Okay, so now I have my UAC, which has with it methionine. That's an amino acid, so it's bringing methionine with it. Now they're going to bind together, and then they're going to move to the next site. So what's going to happen now? They're bound together in the arrival site. They scoot over into the protein site. When I scoot over, I have a new codon that's now in the arrival site. So this is now open for its matching tRNA to come in. Okay? So its tRNA is going to come in, and then this sequence is going to move to the E site. The exit. Everybody with me? Okay. Now something special happens in the protein site, the P site. That little amino acid, the little buddy that was brought in, it's going to stay in the P site. So when these two codons move on, this little guy is going to stay behind. And that amino acid is going to start building with the next one and the next one and the process is gonna happen pretty quickly. So what's gonna happen then is we're gonna start the process of elongation. So elongation is where we continue to have a codon of mRNA enter the A site. When it comes in, tRNA will match up to it. And then together they move to the P site. And don't worry, I'll show you pictures of this to make sense. When they get into the P site, the amino acid gets left behind. And then the tRNA and the mRNA move to the exit site, and then they're going to move out of the ribosome. Okay. Now, the mRNA, as it leaves the ribosome, it's still attached to the chain behind it, so the mRNA is going to kind of dangle there a little. The tRNA, as soon as it leaves, it's going to run around and find its matching amino acid again. So when we're eating and we're metabolizing and we have amino acids, they're floating in the cells, that tRNA is going to go and find its buddy. So for example, UAC, as soon as it leaves its methionine behind in the P site, and it gets out of the E site, it's going to run around and find another methionine. And it's going to grab it, and now it can go back in to the ribosome. Okay, so I'm going to go and find my next amino acid. Okay, so here's our process again. So here we are right here, our second codon, our anti-codon coming in to match up. They're going to match in the A site and then move on together to the next site. Okay, so let's see what happens when they move. Well, when they get close to each other and they bind here, my methionine recognizes that leucine has entered the chat. And when leucine moves over to the protein side, leucine is going to bind with methionine and stay behind. Okay? So what we're going to see is they are now bound. And then that codon, my start codon that brought in methionine, is moved along. Leucine's here, it's going to be dropped off, and then this codon's going to move. And now we have the next codon coming in with glycine. Okay? We're going to continue this process of having anticodons come in and leaving their amino acids behind until we start to get this. Oh my gosh, how cute. It's a polypeptide chain. What's another word for a polypeptide chain? A protein. I'm making a protein. Alright? So I'm going to do this process and make a long chain of proteins. Maybe it'll be short, maybe it'll be linear, maybe it'll be long, maybe it's going to be your secondary folding. Who knows? We'll see what we get. Okay, so this process continues. A, P, E site, matching mRNA and tRNA, and dropping off amino acids. Everything goes along great until I hit a stop codon. So a stop codon enters into the A site. All of these tRNAs are frantically trying to match up with it. They're like, do you have it? Do you have it? Do you have it? They're all running in, trying to match. Nobody can match. Okay, because it's a stop codon. There's nothing that can match it. So what's going to happen now is that codon is going to move to the P site without a match. This is the signal to the ribosome that we're done here. All right, if I go to the P site and I don't have a tRNA sitting with me, I'm done. All right, it's like going to a couples dance without a partner. All right, it's not going to happen. So what's going to happen then is I'm sitting in this A site, nobody can bind with me. I move over to the P site, and the ribosome says we're done. Okay, because nothing's going to match it. So what it's going to do is it's going to release the protein sequence that it's put together. And then my little hamburger bun is going to separate again. And they're going to release that mRNA. So my hamburger bun separates, the mRNA gets released, and my protein gets released. Now, if this was a eukaryote, if I made a protein, I need to package it. Who is my packaging center? Our Golgi apparatus. So this protein is going to have enzymes grab it and take it to the Golgi. We're going to put it in a vesicle and we're going to send it to where it needs to go. If I'm a prokaryote, that protein stays inside and I use it right away. All right? My mRNA strand that I don't need anymore, for us, it goes to the recycling center. Who is the recycling center? Lysosome, yeah. So it's going to go to the lysosome and get broken down. We don't need it anymore. We're done. If I'm a prokaryote, what I'm going to do is I'm going to go and spit it out. All right? So these proteins that we don't need anymore, the mRNA strand, they just spit it out as waste. It's gonna leave the cell as waste. And my process is done. Alright, so I know that was bulky and we spent some time on it, but do you think you could explain this or do this on an exam? Yeah, kind of maybe. I tell you what, I will post tonight a practice sheet for you. And it's a practice sheet where you're gonna just practice translating dRNA and mRNA and tRNA. And it's just kind of a fun one. It's gonna have maybe some fake amino acids, we'll see. But the idea is the same, finding them in, matching them up, and reading them as a sequence. Okay, so I'll post that tonight. It'll be optional. I won't be collecting it, but it's something you can do at home just to make sure that you feel comfortable with it. And then that also lets you come see me if you're stuck. Okay, sound good? Get that posted out top. Okay, so what could possibly go wrong in this sequence? Well, a lot. So let's say that we end up with a mutation. So mutations are where we end up with the wrong information for an amino acid. Now, sometimes mutations can be neutral, which means that if I change one amino acid in a long chain, it probably isn't going to have that much of an impact. Okay? Kind of like if I have a big company and I have somebody else come in and work somebody else's position, it's probably not going to be a huge impact because everybody else is helping and carries the load. So that can be a neutral change. Sometimes we can get beneficial mutations. Anybody seen Spider-Man? Yeah, how cool would that be to swing around on webs? I think that'd be cool. I'm scared to death of spiders, but I'd love to have that ability. That'd be so cool. Maybe I'd be a little less whiny. I feel like being touched is a little too whiny. But whatever, I digress. Okay, so the idea of mutation, something that changes in our DNA, sometimes it's beneficial. Beneficial, there we go. Okay, one of the biggest mutations that was beneficial was this guy. What's this guy? Opposable thumbs. Can you imagine a life where we could not text? I can't imagine trying to hold our phones without the opposable thumbs, or texting, or doing anything. We need these thumbs. This was a mutation. It was a mutation, it was an oops. You can see it in the fossil record. We suddenly had a phalange, a finger that turned more like a thumb, and all of a sudden we could grasp. And once we started grasping, we could start making tools, we could start building. So this was a great mutation, very beneficial. Sometimes mutations are harmful. So a harmful mutation is where something goes wrong, and instead of my baseball glove, I get another. Okay, so something went wrong and I got the wrong thing. Now the way that we can get mutations, it can happen through exposure of what we call mutagens. We've all heard in our life that asbestos is bad for us, right? The reason asbestos is bad is if we breathe it in, where asbestos contacts our lungs, it can cause mutations. It can cause base sequences to get altered. Okay, so that would be a bad mutation. Also, we can have spontaneous mutations. After so many divisions, cells sometimes make an error. We have a ton of enzymes that are always proofreading, but sometimes as the cell ages, mistakes happen. And if we get a mistake, things can go wrong. So that could be a spontaneous mutation. It just happens by chance. Different types of mutations affect us differently. So the first type of mutation we'll talk about is the one that has the least impact. And that's called a missense or a point mutation. Now the way this works is where I'm changing one base. So when I change the base, the word that I wanted, the amino acid that I wanted there, is now changed. So to kind of picture this a little bit easier, I put together 3-letter words. Okay, just like a codon, so 3-letter words, all right? So the correct DNA sequence I want is the cat ate the rat. Okay, that's the sequence that I want. If I do a point mutation, I might sound old English. The cat ate the rat. Why? Because I changed one base. And yes, I know Y is not an amino acid. I'm using this as an example. Okay, I had somebody last semester who was like, excuse me, I thought that a Y. I know. I'm just using it. Okay. Or I could say the cat ate the rat. Okay? Now, changing just one of those, probably not the end of the world. That would probably be a neutral mutation. But you can see where if I start to get a whole bunch of these things together, right? Okay, so this is showing an example. For example, here I have a T which means that my mRNA is going to have an A, which means I'm going to get serine and then a stop when I really wanted glycine. So one amino acid difference. And again, it'll probably be masked, not a big deal. But things can get worse. I can get what's called a nonsense mutation. And this is where it gets a little scary. Nonsense mutations are where the point mutation results in a stop codon. Okay, so a nonsense mutation means when I change the base, I got to stop. And that's scary because the stop codon tells my rest of my sequence that we're not going further. Okay, so instead of getting the cat ate the rat, I might get something like thy cat ate that. And then I hit the stop codon and I never even get to the next amino acid. Now this can be really detrimental if I have maybe like 1,000 amino acid sequence and I get a stop codon in like the first 20 amino acids. So that means the rest of the sequence just drops off. So you see where this could be more damaging, right? It could stop the whole flow of the process. So getting a stop codon, that's starting to get serious. So for example, down here, I wanted methionine, lysine, phenylalanine, glycine. I got methionine and stop. And then that's it. So I'm not getting anything. So I'm getting into a more dangerous situation. The last one I'll cover is the worst, which is the frame shift mutation. This occurs whenever there is an insertion or a deletion of a DNA sequence. Now the way this works is I either stick an extra base in there or I remove a base. Again, it's a misreading. The problem with this is it causes a frame shift. Everything changes. So instead of having the cat ate the rat, I get where I get an extra letter inserted. Okay, so for example, the B here is an extra letter. The B comes in and everything shifts because I can only read 3 letters at a time. So B comes in, here's my word, C-A-T, but because the B is here, the T got pushed to the next codon. And then so C-A-T, it should be A-T-E, but because that guy got pushed, now the E's over here. Everybody get it? So everybody gets shifted by 1. The reason this is a problem, I'm going to get an entirely different chain of amino acids. So what I wanted was methionine, lysine, phenylalanine, glycine, and stop. I got methionine, lysine, leucine, alanine. I got all sorts of different amino acids. This is where I would be getting like a baseball glove instead of a mitt or vice versa. I'm getting a functional product, which is not what I wanted. Okay? So a lot of times we'll see this in things like genetic mutations where things aren't working right. We also see this in things like UV light exposure. So UV light, we know if we get too much UV on our skin, it can cause skin cancer. The reason for that, if I have 2 T's together, the UV light a lot of times causes these 2 T's to stick together and they're read as one base. So I essentially lose one of them. So then everything frame shifts. We call that a thymine dimer, they stick. And now they're being read as a single instead of a double.